Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but you're welcome to type in your questions for a speaker in the question and answer window on your screen. After the presentation, I'll be asking your questions to our speaker Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Deborah Pascoe from Culture Biosciences.
Hi, everyone. It's a pleasure to be here with you today to share some of our recent work in CHO cell culture. I joined Culture Biosciences last year as our Vice President of Operations, and in that role, I'm responsible for our BioProcess operations team, our client success team, and our business operations work. Before we get started, I wanted to give a shout out to my colleague, Michael McSunis, who led the CHO development work that you're going to see today, as well as to say thank you to our colleagues in hardware, software, and our biooperations team who did so much to generate these results. So some brief background on the company, because we're well known in the microbial space, but less so in the mammalian cell culture space. Culture is your bioprocess lab in the cloud. I do want to assure you we have a very real, very physical lab here in South San Francisco. But as far as the people we work with are concerned, everything is interfaced through the comfort of your own lab or office at your laptop. So we really are in the cloud from that perspective. Our focus is really on the execution of bioprocess experiments. We've developed our own 250 ml bioreactor system, which you can see an image of here, that we build in-house. And we've continually ramped up our capacity over the last 2 years, working with clients on microbial cultures. We really see ourselves as an extension of your lab. You focus on the experiment design and the analysis, and we focus on the execution piece in the middle. The way that most companies work with us is in an ongoing guaranteed capacity model. So what this looks like is that you sign up for a specific number of bioreactor runs per month or a minimum number of runs per month, and then each month you send us the experiment designs, we communicate back and forth to make sure we have everything well understood and we're going to execute it correctly. We execute it in our labs, and then you get the data back online as well as the samples, and you analyze those and focus on moving your program forward. For companies with a significant internal bioreactor capacity, this means that you have the flexibility to add runs when something new comes up or that inevitable challenge that comes up in every project. Something new happens and you realize you need to run more experiments. Or you can add additional programs without affecting the timeline of your overall portfolio so that you have this flexible capacity on hand. For companies with limited or no bioreactor capability, having immediate access to bioreactors can completely change your approach to your program. With us, within weeks, you can be up and running your tech transfer experiments, and then from there we go into this ongoing guaranteed capacity model. So this differs from a traditional CDMO in a couple of ways. Really, you don't just throw it over the fence to us and we work on it on ourselves and then give you something back and you don't know how we got there. You're involved in every step of the way. So you have the control of the experiments, you're designing those. You can see the work in progress. You can watch the trends of the bioreactors as they're running and see the samples as they're taken. And you can get started on these experiments in less than a month in most cases. So we have, instead of having to wait the 6 to 12 months that it typically takes to bring new capacity into your own facility, you can get up and running quickly. And then speaking of the facility, you don't have to worry about maintaining the facility, the CapEx associated with bringing in those bioreactors, or hiring the team to run those bioreactors, because we all know that it takes certain skills and expertise and a lot of practice to be able to run these bioreactors well. So I think I've covered a lot of the points here, but I'll keep moving. But I did want to share this picture of one of our bioreactors because it's a nice close-up. I will say this is definitely not a CHO process. We don't need agitation of that rates for a CHO. But again, we've had many customers that we've worked with over the last few years for bacterial and yeast and even fungal cultures, and now several early CHO cell clients. And we're excited to expand our mammalian cell culture work this year. So here's a couple of examples for experiments that we can do for CHO. A really straightforward way to get started is clone screening experiments where you're running one process or one or two processes and screening a large number of cell lines through those. And then moving into baseline process development, the first process you want to be generating your early material in, getting that up and running, or developing that first process so you have it ready to go. And then, of course, from there, progressing through media and feed optimization, process parameter optimization, all the way through, and these studies become larger, you can be running DOE studies, and all the way through characterization and validation. Because we can run so many samples at once in our modular system, we can save time and increase your confidence in the results by running more conditions and replicates at a time. A quick technical review of the system. Our automated bioreactor infrastructure builds on the best features of comparable off-the-shelf systems. What you don't see here in this figure that I'd like to highlight is the automation and measurement capability. We run with automated feed triggers, so we can feed based on different process parameters or different characteristics occurring at a time. We can run simple or dynamic feeding strategies. All of our feeds are delivered by peristaltic pump with scale feedback control. So this means, especially in a case like CHO, where you can run for multiple weeks and you may see some difference as the tubing gets older in those pumps, we can still adjust for that and make sure we're feeding correctly. We also have all of our gases on mass flow controllers, and so we're tracking all of the inputs and outputs of the bioreactor, both liquid and gas, which lets us publish a mass balance on every single run. And we recently did a retrospective of the last many hundred microbial runs, and we found that of those, so wide variety of processes, about 75% of those were within 3% closed or better. And one other thing to note is that each of our bioreactors is fed completely independently. So you can have a different feeding protocol for every run, as well as for every case, for every firm, as well as different feed compositions as well. The other component of our technology is our software, which is our cloud console. So console is where you'll enter your process designs and the other details, where you'll access those live data online. You can even view images of the reactors running. We have a camera on every reactor, so you can see what they look like in process. And then at the end of the run, this is where you'll access data from all of the cases or all of the things you've ever run before. And you can choose to analyze your work here in console, or you can choose to take it outside. So most of our customers use console for their analysis. We upload offline data, you upload your own offline data, and so you can have it all in one place. Or if you already have extensive systems externally that you want to run in and add the data to these, you can extract everything through our API and analyze it there. Again, as an extension of your lab, we want to do what makes the most sense for you. We're happy to go either way. One feature that we recently added to our console is what we call workspaces, and we're really excited about this. We use these internally all the time. A workspace is basically a setup where you'll put all your favorite graphs and analyses that you like to do and have those ready to go. So with every experiment, it's just a few clicks to generate all of those plots or to look at things across experiments. You can also share these with colleagues by giving them a login as well. There is no per seat charge. You can have as many of your colleagues able to log into console as well. And so now we find that people are instead of like extracting graphs and making PowerPoint presentations or group meetings or things like that, especially now while so many people are working remotely, it's easy for everyone to just log in, look through the plots, discuss it, and move on with the planning for the next experiment. So, and again, so there's no per seat charge for that. There's no charge for the software. It's all just part of the service. So a quick summary, I think, of what we've gone over here, what this takes off your plate. You provide the process parameters and the vials, send those to us. We do the hard work of building and prepping the reactors, maintaining the facility, running the firms, taking the samples, taking down the vessels at the end, and while you just monitor the experiments in real time. And then at the end of that, you can add your own offline analyses, or in many cases, people don't have to do that because we're also capable of running product assays for customers. But you'll add any other analyses, and then you'll look at your results and decide and move forward. So let's move on and look at a few different cases from our recent CHO cell experiments. So I want to highlight here also that we have, what we're going to show today is, has a lot more extensive information in a white paper we recently published. So if you go to our website, culturebiosciences.com, and click on resources, you'll find the white paper that describes these experiments in a lot more detail, as well as spec sheets and other different pieces of information for us. So this work was in partnership with an early client and working on CHO cells. And they sent us two different cell lines. So here we're calling them line A and line B, and they make two different products. We ran them, first of all, in a process we'll call, logically, process 1. And we ran them both the same way, except they knew that they wanted a subtle difference at the beginning for each cell line. Cell line A started at 7.3, cell line B started at 7.2. And these were, but they both shipped to 6.9. So there's just that small difference at the beginning per cell line. Otherwise, it's completely the same. So this was a great way to do an initial test of customer cell lines in our system. So we got going with process 1. By the time we'd finished those experiments, the customer was ready to send us a slightly revised process, which we're calling process 2. Process 2 really only differs in the inoculum train. That's where the key difference, well, that's where all of the differences are between process 1 and process 2. So this let us look both at differences, even the subtle differences you might pick up from how you've made changes in the inoc train. We were able to see that in our systems. And it also let us compare from our 250 mil scale to the customer's benchtop scale. So we're able to look within systems in this experiment, as well as looking across different scales. So here are some plots we've pasted straight from console. So this is what it looks like to you in the analysis, not in a grid like this, but these are the kind of plots that you'll see. With the webinar set up today, we couldn't do a live demo, but I have plenty of colleagues who'd be happy to walk through a demo of console with you. So just to orient everyone to what's here, on the left is cell line A, on the right is cell line B. The red and the blue are process 2, so the newer process. You see it grows a little bit more slowly. The different shades within each color mean they were separately run independent experiments, but of the same process. And then finally, the top plots are our viable cell count, and the bottom plots are the viability, and that was all run by Vicell. So this is really a straightforward look at replicates for several cases over several experiments. You might also notice that process 2 performs in a way that we probably think about as being more favorable, better viability, longer in the run, and more sustained cell count later on. And part of this is, we think, is due to the process changes, but we also, between running process 1 and 2, did additional work on our control systems. So we've made some adjustments to our process control algorithms and tuning for pH and DO. And we think this has also helped get that even better reproducibility that you see there. The next plots are titer on the top and then lactate on the bottom. And we chose lactate concentration as a representative metabolite because it's one that people think about a lot and care about a lot. So you can see the titers are highly reproducible. These lactate levels, they're all low and pretty consistent. They have different axes here on these plots. They're all consistent. We did see divergent behavior with cell line B in process 2. You'll see the blue lines, but the client also saw this in their reactor system. So this is a job for more process development to tighten this up again and make this much more reliable and reproducible, but it was very consistent across scale. So in that regard, we were comfortable with that. So it's great to see this consistency both within the cell lines and across the different cases, and to be able to see these differences that came from the inoc train changes as well. I do want to add there's a slew of other nutrient metabolite control information in the white paper, pH, PCO2, DO, glucose, glutamine, glutamate, ammonia, osmo, all the usual suspects are there in the paper. We just didn't want to hit you with dozens of graphs in this, again, in this slide format. But I think we've shown here that we have this consistency across these two different cases within our scales. So within culture system. So let's take a look then at comparing process 2 to customer benchtop data. So here now we're looking across scales. And again, the red and the blue are process 2 run at culture. And then the gray and the black lines are run at the customer site. And so we do see here things are highly reproducible within our cases. We did see a better viability run at culture, and you especially see that for cell line B. And we think that this might be due, again, we mentioned we made some of these control algorithm changes. We think that the better viability may be due to the fact that we had better pH and more consistent pH control early in the run and later in the run. We're in very different regimes for the culture, but we had much tighter pH control at those points. And so, but again, what we see here is that they overlay well. If we move into titer, again here at the top, and lactate at the bottom, these are the same curves. It's just slightly different axis on the one on the left, so it looks lower. You'll see here for cell line B on the right in blue, we had this variation, and you also see that in the customer scale, that later in the culture, they diverge slightly. I did mention, in thinking about the improvements we continue to make to our control algorithms, I do want to mention that this is the type of thing that we want to think about and manage closely with a client. In some cases, you may want to take those learnings from what we're doing and transfer them to your own system and move forward with those levels of control. But in other cases, that might not be doable at scale. And so in that case, we should work together to loosen up those controls to make sure that what we're running is representative of what you'll run at scale. And again, we're happy to do whatever makes sense to you. The point is to get this work done well and to be able to scale it up well. So overall, this is a high-level view of the success of these early experiments. And again, I'd encourage you to go look at the white paper at culturebiosciences.com under the resources tab, along with the other publications we have there. I'll leave you with this photo of our team. This is from the very early days. We can no longer take a picture with everyone with no masks and close together. But rest assured, our team is much bigger now. And want to mention, we're on site 7 days a week. So we have both a day shift and an evening shift. So we have almost around the clock coverage for sampling, monitoring. And again, we can monitor remotely. You're also monitoring. But we have 2 shifts a day, which means you can do a lot more extensive sampling than cases where you may have your process development scientists who work 9 to 5-ish Monday to Friday, and so you don't have the same level of coverage. But with that, I want to make sure we have some time for questions. So I'm going to hand it back over to Leah for questions.
Okay, great. Thanks, Deborah. So the first question is, how can your customers be confident the process will run exactly as intended?
Yeah, because it's always, there's already so many things that can change or go wrong or be different across systems, right? So in the contracting phase, we work hard on defining the tech transfer requirements. So every process should go through a tech transfer stage before we move into that ongoing guaranteed capacity mode. So we work with you to define what tech transfer success looks like. And we don't leave that tech transfer stage until both parties agree that success has been met. In some cases, tech transfer might be, we haven't run this very often, we just want to see it goes at your site and we'll move forward. In other cases where you have a lot of process history, we can put statistical quantification around that. You also, on an ongoing basis, have a client success team representative who works closely with you. And within our BioProcess team, we have a program lead who's responsible for knowing the ins and outs of the process and knowing it well and keeping track of it.
Okay. And can you measure biomass, BCD, or TCD online during runs?
Yeah, so we are measuring, we have multiple measurements, and we're happy to put in different probes or different systems depending on the customer need and what they want to analyze. We're certainly capable of doing ViCell counts, manual counts. Ideally, we don't do manual counts, and viable cell density measurements throughout the run.
And what type of companies do you work with? What types of processes can you run?
Yeah, we have so far worked with companies across the spectrum. So everything from food products, agricultural products, synthetic biology companies making interesting novel materials, as well as pharmaceutical and nutritional focused companies. We also, so sort of the types of companies or the types of products, we also worked with a very different mix of microbes, so bacteria, yeast, fungal cultures, CHO cells. And as I mentioned, we're looking forward to expanding to other mammalian cell types this year. And then the other piece of that, I guess, is company scale. So we work with clients of very different scales. We have a significant fraction of companies who have very little to no bioreactor capability, but we also run very large studies for other companies who themselves have a decent amount of capacity. And it's fun to work with all of those different scales and different people, whether it's the one product they're really focused on or it's part of a larger portfolio and you're helping work with a really capable team getting work done.
And can you tell us a little bit about the cost of the service?
The cost of the service is going to vary depending on the length of your culture because, obviously it takes more or less time in bioreactors means we can or can't run other companies and it's a different amount of capacity. But I would encourage you to look at our website because we actually have a cost calculator on there where you can look at whether buying your own bioreactor is right for you, working with culture is right for you, where the trade-offs would be for that. So again, it really comes down to duration and the number of runs that you want to do. But I would encourage you to look there for some sort of first examples.
Okay, great. So we have time for just one more question. There are a number of questions here, but if we didn't get to your question, we'll be passing those along to Deborah and she can follow up with you directly. So don't hesitate, keep typing in questions. But the last question for this presentation is, what if a customer has a proprietary cell line that is too difficult to handle, so tech transfer can be problematic? Then can Culture Biosciences set up a small system for 10 to 20, 250 milliliter mammalian bioreactors in the customer's R&D lab?
At this point, we run everything in our labs, so it's a complete service. And so part of the efficiency in us being able to do this work is we're building the reactors, we can service them, maintain them. So at this time, our model is that we run everything on-site in our labs. Of course, if it's difficult to run, we will partner with you on getting that up and going. We want your input and insights through the whole process. So in that scenario, we'd work closely with you, of course, during the tech transfer phase to get that up and going.
Okay, great. Thanks, Deborah.
Thank you. Thanks, everyone, for being here today. It was a pleasure to share some of our recent work with you.
And the recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.